Welcome back. What I want you to put on underneath the gamete in the sentence you just wrote down is the title for this concept and this video, which is incomplete dominance. Incomplete dominance is the title of this video, as you probably saw in the file folder for this video. Incomplete dominance is similar to the Punnett squares that we have been working with already, except that there's not a single dominant gene. So far what we've had is that when you have, say, an H for a gene, the dominant trait is all that you see when there are only dominant genes. If you only have recessive genes, all you see is the recessive trait. And so far everything we've seen is that if you have a dominant gene, a capital letter, and a recessive gene, you get which trait? Dominant trait. Now, this is a little bit different because for incomplete dominance, what occurs is that, and let's use colors of flowers. For instance, in your book, it talks about a red and white flower. And you'll see in incomplete dominance and codominance, which we talk about next, what's different is you use two capital letters for these, because there's not a single recessive trait that won't show up. Instead, when we have red and when we have white, if we mix two white flowers, you should be able to guess what color all of the offspring will be. They'll be white. And if instead we mixed all red flowers, you would have all red offspring. What happens though, when you mix, say, a red flower and a white flower, when you have that heterozygous mixture of one letter, of two different letters, and instead of a capital letter and a small letter, for incomplete dominance, and if you ever see us mixing two different letters, then you know, instead of having a dominant trait over a recessive trait, we have something called incomplete dominance, which incomplete means not complete, obviously, and complete means finished, or completely taken over would mean that you've taken over everything. A completely dominant trait means that it is dominant over all of the recessive trait. What happens here is that instead, neither of these letters becomes completely dominant, and instead the two letters mix. So this, you end up with the genotype of red and white, or I guess the genotype of R and W for every flower. So a 100% genotype of RW, but the difference here is that RW, instead of equaling red or white because they're two capital letters, you get a pink flower. And the same thing, we saw one of these on the test that had black hair and blonde hair, and incomplete dominance in the middle was brown hair. So what I want to do is on your worksheet, on the page you're taking notes on, we're going to give you that a B equals blonde hair and an L equals black hair. What I want you to do on your page of notes is a Punnett square of a homozygous black hair person and a homozygous blonde hair person. So homozygous blonde, and you've already got the key here. The difference will be that when it's heterozygous, it will be brown or mixture between the two. So I won't tell you what heterozygous is. Hopefully you just saw in the last one. You can go see what the last example is if you need. But this is brown when it's heterozygous. So we have a gene for blonde and we have a gene for black hair, but we have the phenotypes of blonde, black, or brown. So I want you to mix a homozygous black-haired person with a homozygous blonde-haired person. Do that on your sheet for a Punnett square, and you can pause this till you're done. So you just finished the Punnett square with B and L for black hair and blonde hair, or blonde hair and black hair, the way that it was. What I want you to do is underneath that Punnett square, we're going to do another incomplete dominance Punnett square. Instead of mixing only one, basically homozygous genotypes together, I want you to mix a heterozygous pink flower with a homozygous red flower. And what I'm going to be looking for, other than the Punnett square, so you've got the key here, copy that onto your notes, then you're mixing a heterozygous pink flower with a homozygous red flower. I want to see that Punnett square, and I want you to tell me the probability, or that phenotypic ratio, the probability that a flower from these two plants will look red, solid red. What is the probability that a heterozygous pink flower and a homozygous red flower will make a red flower?